Hi everyone. In this video, I'll talk about Golgi apparatus and I'll take you through the structures to the functions of Golgi apparatus. Now, if you watched my other videos, you know that the endoplasmic reticulum of a cell is just like a factory where the proteins are formed, proteins are folded, they're packaged, and they're quality checked for distributing to the other sides of the cell. Now, this factory is associated with a warehouse, and that warehouse is actually the Golgi apparatus. So, clearly, the Golgi apparatus could be understood as a warehouse of a cell where several products are packaged, sorted out for delivery to distant locations inside the cell or maybe secreted outside the cell. Now, just like a warehouse where trucks are taking the parcels and the products for delivery it to several part of the city, from Golgi apparatus there are specific vesicles going either towards out of the cell for secretory products. Maybe some of the vesicles are going towards specific organelles like lysosome. Some of them might go to mitochondria and like different other locations inside the cell. Now, this process is well coordinated by several intermediate players, and for Golgi as well, this process is well coordinated. Now, imagine that this is the endoplasmic reticulum. There are several vesicles which are budding off the endoplasmic reticulum, is fusing with a cis Golgi, where there are intra-Golgi transport and the products are modified, labeled, and ready to delivery to several locations inside the cell or outside of the cell. We would look at how this sorting process works inside the Golgi, how the transport is selectively delivering the product inside the several locations inside the cell or maybe outside the cell. And also, we have to understand this process of sorting and delivery that is taken care of by Golgi is pretty complicated. Imagine selective receptors that need to be delivered to the apical surface, or some ion channels, uh, or some protein molecules that has to be delivered into the basal surface of the cell. So how does Golgi ensures that the proteins that need to be displayed on the apical surface are selectively delivered to the apical surface, not the basal surface, and vice versa. So this kind of stringent decision and stringent sorting kind of activity happens in the Golgi, and that is why Golgi is such an important structure. Now, if we imagine Golgi as a post office of a cell, then you can understand all the parcels or the letters that need to be distributed to several locations inside the city that has to be address tagged, right? So the proteins inside the Golgi also need to be tagged with a unique marker, and the tag turns out to be a N-linked glycosylation tag. Glycosylation simply means attaching sugar to something. Now here, a complex sugar or oligosaccharide is attached to the newly synthesized protein, and this attachment of the tag starts in the ER, and this tag is work like an address barcode, which would help the sorting machinery or the vesicles to deliver it into specific locations. Later part of this video, we will take a specific example to look at how lysosomal hydrolyzed enzymes are selectively delivered to the lysosome and not any other locations inside the cell. Now, first we should ask that where does this glycosylation taking place? It turns out glycosylation starts in the endoplasmic reticulum, but quite a lot of part is happening in the Golgi networks. In the Golgi networks, there are basic functions which ensures that several mannose residues are chopped off from these basic tag. There are other enzymes which ensures several N-acetyl glucosamine is added to this tag. Several other transferase enzyme transfers galactose, sialic acid, etc. in the trans-Golgi network. Now, this specific modification ensures which protein is going to be delivered to which location. We take an example of delivery of hydrolyzed enzyme into the lysosome. Now, hydrolyzed enzyme has the basic tag. One modifier enzyme, known as phosphotransferase, attaches a phosphate residue on the mannose. And this mannose 6 phosphate modification is going to be recognized by mannose 6 phosphate receptor. Now, mannose 6 phosphate receptors quickly work like a sorting signal. They allow to bind the adapter protein along with it, and they attract triskelion molecules which would ultimately form a clathrin coat and allow the clathrin coated vesicles to bud out from the trans-Golgi network. Later, using specialized molecular motors, which are just like trucks, these cargoes would be delivered to specific locations inside the lysosome. Now, once they are reaching the lysosome, they're going to fuse with the lysosomal membrane. Inside the lysosome, the pH is different because lysosome also has proton pumps that always pumping in proton, that making the lysosomal interior to be a lot more acidic. In this acidic environment, the interaction between the mannose 6 phosphate receptor and the mannose 6 phosphate signal is broken. There are other components which would chop off this mannose 6 phosphate signal such that the enzyme that reached the lysosome cannot go back. Once it is done, then the empty trucks, that means the vesicle, is going to be carried back and it might fuse with the Golgi with the help of a retrograde transport. That is how specific proteins or specific enzymes are delivered to specific locations of the cells using the help of specialized sorting mechanism inside the Golgi apparatus. And also, this is very important in terms of proper segregation of the products. Improper transport of this lysosomal hydrolase might cause diseases like lysosomal storage disorders, which could be sometimes detrimental. So that is why Golgi apparatus is so important. So in this video, we looked at Golgi apparatus from a structural to the functional point of view. If you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.